for our project, we did the snack food A5J, sorry. And the goals of this project is to develop and evaluate isolation slash separation methods that best support a mass-based quantification of lipids in snack foods, devising and running tests to verify the absence of non-lipid food components likely to interfere by adding mass with the lipid quantitation with will be important. Subsequently, your best approach is applied to quantitate the fat in an assigned snack food, applying a aforementioned test to confirm only lipid is being measured. Finally, your group will compare results with another group within your lab session, analyzing the same snack food to further gauge the reliability of the methods. And again, our sample food was A5J, which we observed was the brand, the Pringle chips. So for group A, we began with the isolation of the lipid, and we first just crushed up the A5J snack and used a mortar and a pestle to weigh 1 gram of the sample. Then we transferred it into a centrifuge tube, about 1 gram, and we watered it down with 2 to 3 milliliters of acidic water and cyclohexane, and then placed it into the centrifuge tube for 3 minutes. And then after removing it, we used a pipette to remove the top liquid layer, and then we placed it into a beaker and then on a hot plate and heated for about 5 minutes. After removing the liquid, we weighed it to calculate for the mass of the isolated sample, and then we began our control tests, which is to confirm that the procedure was done correctly, and we used the same steps in our assigned drink, and then we performed it on the corn oil. And we again added the corn oil into a centrifuge tube and put in 2 to 3 milliliters of cyclohexane and acidic water. And then we placed it into the centrifuge for 3 minutes. And then once it was done, we transferred the top layer into a beaker and heated for 5 minutes. And again, after it was done, we waited, and then we weighed it to calculate the mass of the isolated sample. And then to perform protein tests on the control test as well, and then the iodine test. So the iodine test is to confirm that the isolated lipid is free of starch, and we first diluted it with the 2 solvents, acidic water and cyclohexane, and then we added 1 drop of the iodine test solution and stirred it. And then if the solution is a yellow-orange color, that means that there's no starch present, and if the solution was a black or a dark blue, the solution still contains starch. And in the protein test, we added 1 to 2 milliliters of salt water into an isolated lipid solution and again stirred. If the solution is a foggy, bubbly texture, then the solution still contained the protein, but if it was clear, then it did not. And after performing 2 trials, we got this data. So in trial 1, we had a mass of 1 gram, which we weighed in the beginning. And then after heating it, we got the grams 0.231 grams for the first trial and 0.109 grams. And then we calculated the percent by fat weight, which is 22.87% for our first trial and 18.48% for our second trial. And we repeated the same steps for the control test, which we got 2.21 grams when we first weighed it for the first trial and 2.288 for the second trial. And then after heating it, we got 2 grams exactly, and then 5.181 for trial 2. And then we calculated the percent fat by weight, which is 90.5% and 82.73%. And then we just divided that to get the average percent fat by weight, which is 56.69 for the first trial and 50.61 for our second trial. And then here are our calculations. So to get the percent fat by weight, we just divided mass of fat and the mass of sample, then we multiplied it by 100, which is how we got the percent fat by weight for our assigned food and our control test. And we got our average percent fat by adding the 2 percentages of our assigned snack food and then the control test and divided it by 2.
All right, so for Group B, it was uh, Ziran and I, Jacob Civil. And so for our isolation outline, you pretty much want to start with crushing and separating 1 gram of the A5J snack food, and then add to the centrifugal tube, you want to add 2 milliliters of methanol and then shake it for about a minute. And then you'll add 2 milliliters of cyclohexane and shake for another minute. Then after that, you want to run it in the centrifuge for 3 minutes. Then after running it through the centrifuge, you want to remove the liquid, the top layer of the liquid, and then you want to place it on the so you take that top layer, put it in a beaker, and then on that beaker, put it on a hot plate and turn it to the 3 heat setting, and then wait for it to boil. After it stops boiling, which you'll know when there's no more condensation on the side, you'll take it off and then recalculate the weight. And then you'll add 1 milliliter of cyclohexane. Then you'll perform the iodine test, which is just like in that little bottle, and you just do 1 drop. And then you'll do the perform the protein test, which is just putting like 3 to 5 drops of salt water in there. And then for our control test, this is kind of what we're basing our results off of. We did a centrifugal tube with 2 grams of corn oil, and then we'd added 2 milliliters of cyclohexane, and then shook it for 1 minute. We added 2 milliliters of cyclohexane after that, and then shook it for another minute. Ran the centrifuge for 3 minutes, separated the top layer into a beaker, and then placed that beaker on a hot plate, and then turned to the heat setting 3 and waited for it to boil. Then after it stops boiling, we recalculated the weight, add 1 milliliter of cyclohexane, and then just did the iodine and protein test just to make sure that it was all similar. And then this should prove that our test is effective because it doesn't. It should all be like similar. Like it shouldn't pass the iodine or protein test. Like both of them shouldn't, and they should be similar, like concentration or like percent fat by weight. Next slide. So for the iodine test, we just we did it after we boiled the cyclohexane off and calculated the weight, and then we mixed like 1 milliliter of cyclohexane, and then just did 1 drop of the iodine and swirled it around for about 30 seconds. And then if it stayed orange, we knew it was starch free, but if it turned black, then it means that it had starch in it. And then for the protein test, after we did the iodine test, we just got 3 to 5 drops of salt water, and then swirled it around again for like 30 seconds. And then if there's no bubbling, then um, there isn't protein. But if it starts to bubble, then there is protein. Yeah, okay. So here's our results. We did 3 trials. Each of them we did 1 gram with a snack. Then the mass after running through the outline, we got 0.805 grams for the first one, 0.815 grams, and then 0.785 for the last one. So this gets us to the percent fat by weight is 80.5, 81.5, and 78.5 for an average of 80.17. And all of our trials passed the iodine test and then passed the protein test, and then it's not on here, but our control test also did pass both of those. So here's the calculations that we did. So to get the percent fat by weight, we just did the mass of the fat. So after we boiled off all the cyclohexane, and then we did the mass of how much chip we started with. So it was 1 gram. So like this first one, for example, we did 0.815, and then we divided by 1, and that's how we got the 81.5. And then for the control test, it's the same thing. Just we started out with 2 grams, and then um, we divided it by how many we had afterwards, after boiling off the cyclohexane. And then for the average, what we did is we did all 3 trials, what we got the percent fat weight to be, we added them up and then divided it by 3, and that's how we got the 80.17. So this is the comparison between Group A and Group B. So we all started roughly with 1 gram, and then the mass of the isolated lipid, Group B had more afterwards, and then Group A had less. And so then that means that the percent fat by weight would be less for Group A, and then for Group B it was higher. And then for the mass of control test, we did roughly around 2 grams, both groups. And the mass of the lipid isolation, it was about the same, around like 2. We got 1.64. And then the percent fat by weight, yet again, it's all similar, like 82, 82.73. For the iodine test, all of ours passed, and then there was only one exclusion for the protein test, and one of them failed, but the rest of them passed. So recommendations to improve the analysis. So we think that we could find a better way to separate the top layer out of the centrifugal tube, maybe by just using like a coffee filter. Just because we found that when we were running our test, that there were still like tiny little pieces of food snack that we accidentally brought up with it. But yeah.
And then our reflection from this project is we were able to acquire new information, which we could apply it to working in a lab in the future. For example, using a heating plate to boil off a product or having to perform tests to make sure our product was free of starch and protein.